Well, just as a startup disclosure, I do not consider myself an expert in the field of cell and gene therapies at all. We had an excellent speaker lined up for the reflection on the workshop who unfortunately had to cancel her participation 4 weeks ago because of medical reasons. And then we had an excellent substitute who unfortunately had to cancel her participation as well 4 days ago. So I'm here as a kind of second-hand substitute trying to give you a nice overview of what happened at the workshop, but I'll do my best. The workshop was held on November 29 and 30 and actually consisted of 4 sessions spread over the 2 days. And I'll go over the 4 sessions one by one. The first session was dedicated to the biology and the technology related. Trying to move the slide. Yeah, here we are. Just as a reminder, T cells are a very essential part of the immune system. Their killing function is triggered by signaling through the heterodimeric protein T cell surface receptor that you can see here at the bottom part of the slide on the left. This is the TCR. This TCR is able to recognize and bind to a combination of peptides found in the groove of a molecule called MHC that you can see in the top, which is part of the tumor cell in this case. This is one of the tissue protein type proteins that is expressed on the surface of all nucleated cells in our body. So that includes the tumor cells. Binding to the receptor leads to a signal to the T cell and activates this T cell. This induces a clonal expansion of T cells that all express the same receptor. And they can differentiate and secrete cytokines and enzymes that lead to the death of the target cell. Importantly, T cells memory persists in the body for the rest of the host life. So if T cells can be manipulated to recognize cancer cells, they can become an army against the cancer cell involved, an army that will persist long term. This manipulation is performed extracorporeal, and that's shown on the right side, after collecting T cells from the blood of the patient. In a laboratory, after engineering of the cancer cell proteins, the cells can be cultured and expanded, and then reinfused into the patient. Once back in the body, the cells travel to the tumor site, recognize the tumor cells, and kill them. There are various laboratory procedures that are applied in T cell engineering. The most commonly genetically engineered T cells in clinical use currently are the chimeric antigen receptor T cells, or CAR T cells. There are different kinds of receptors that can be introduced into the T cell. These receptors are based on fragments of an antibody. This has the advantage that the antibodies recognize antigens on the surface of cells that have not been processed and presented by the MHC. This in turn means that T cells expressing a chimeric antigen receptor can be used in any patient, regardless of their tissue type. The majority of current clinical trials in T cell therapies are in hematological malignancies, at least in cancer, and are involving the application of CAR T cells. But also natural killer cells are used for this purpose. And the advantage of using such natural killer cells is that they can be derived from a high variety of sources, including healthy donor, peripheral blood mononuclear cells, cord blood, placental blood, induced pluripotent stem cells, embryonic stem cells, and even NK-derived cell lines. Now, this brings us to the clinical development that was discussed in the second session. In the regulatory environment, both EMA and FDA now have specialized teams for assessment of dossiers on those advanced cell and gene therapies, the ATMPs. At EMA, this involves the Committee for Advanced Therapies, and at FDA, it involves the Office of Tissue and Advanced Therapies, or OTAT, within the Center of Biologics and Evaluation and Research, CBER. Both of the regulatory agencies have issued specific guidances that help applicants of dossiers in finding their way. Importantly, the recently activated EMA clinical trial application regulation, only activated a couple of days ago, involves a harmonized procedure for EU with a single application towards a portal located at the EMA. There will be a coordinated multinational assessment with a rapporteur. Yet EUA member states do retain authorization and oversights. While ATMPs are relatively novel, already over the last 9 years, 19 ATMPs have been approved by EMA, including 4 CAR-T products focusing on hematological malignancies. And similar numbers are achieved by FDA. The past focus has been on single-arm studies in areas of unmet medical needs and rare populations. The rareness of those populations could cause attrition issues and screening exhaustion, but also could create operational challenges in manufacturing and therapy delivery. Interesting developments, such as the so-called parent-child protocol development strategy that's shown here, allow to develop multiple technologies in a faster and more cost-efficient way. In the first-in-human protocol, master protocol on the left, there is a platform protocol consisting of a core and multiple independent sub-studies focused on dose, early safety, and multiple mechanisms of action. On the right of this slide is the definitive data master protocol that has the same structure but is focused on proof of concept and on registration. And the third part of this initiative is on the bottom is a single long-term follow-up study to which all patients from all sub-studies and all agents that are involved transfer following the end of their study treatment. This approach provides an ideal framework for cell therapy development. In practical terms, it allows parallel examinations of multiple assets in a single master protocol with cross-referenced INDs. The challenges of developing an academically led clinical protocol and program were outlined by Professor Hudacek from the University of Würzburg using the example of the so-called Caramba trial. This myeloma trial is financed by a grant from the EU Horizon 2020 program and involves 6 European countries. It is supported by Myeloma Patients Europe, which ensures a patient-centered focus. It only took 3 years from the reporting of the very early preclinical data to the entry of the first patient into the clinical trial, which I think is a formidable achievement and confirms the feasibility of this academic approach. Now, this brings us to another very important element, patient access, which was the topic of the third session. After marketing approval, there is a social responsibility to have a broad geographic worldwide patient access to CAR T-cell therapy. This is facing us with new challenges. First, T-cell therapy is different from the more usual drug therapy that we are applying. It is not a daily or weekly repetitive treatment, but it involves a one-time procedure. And this obviously requires different approaches to ensure patient access. Second, most studies on T-cell therapy up to now are small, single-arm studies with major limitations in the translation of their outcome into the real world. The absence of such definitive evidence for clinical use is a serious drawback for HTA appraisal. And if we then look on the existing, much less expensive and more simple standard of care treatments, there is a major inequity in patient access, even at the pan-European level, as is exemplified in this slide from Kate Morgan's lecture on lenalidomide and Imovit in multiple myeloma patients. Many EU countries are many years behind the ESMO standards, unfortunately, and they do simply not have the experience nor the hospital capacity to provide simple standard of care, let be complex CAR-T therapies. Digging somewhat deeper into the HTA process, Professor Karen Uil explained that the ultimate aim in this process is to ensure affordable and equitable access for all patients to effective therapies in a sustainable manner. There is an emphasis there on the words affordable and effective. The HTA process involves the assessment based on available evidence, as shown on the left side of this graph, and subsequently the appraisal, taking several other aspects into account for balancing. Each EU country decides for itself in this HTA pathway. There is no central EU procedure. All countries struggle with the same problem, that the current medical systems are financially not sustainable. In this landscape, and taking into account what is spent on one patient cannot be spent on another, which are the so-called opportunity costs, the per patient cost for CAR T-cell therapies that amount up to 300,000 or 400,000 euros create a true challenge. They can likely not be covered within the allowed maximum yearly budget growth of 1.2% for medicines. So a leading question that countries face is how to reduce their spending rather than how to add expensive treatments. To address this issue, value-based pricing has been introduced. This includes factors such as cost per quality of life year gained, pay for performance, volume price arrangements, and more recently, the coverage with evidence development, or CED. CED is particularly applied for drugs or therapies for which adequate large data sets are yet lacking. And this involves the gene and cell therapies. Clearly, in actual practice, patients' access has been by far guaranteed within the EU, and there is a long way to go to improve. The requirements of evidence providing large studies for patient access led into the discussion in session 4 of the workshop that was dedicated on future study aspects. Speakers discussed the potential of using registries or other sources of real-world data as an external comparator arm that in theory could establish context for single-arm trials. In assessing external comparators, one should differentiate external controls that include adjustments of data at the patient level from benchmark controls that exclude such adjustments. The good message is that there is increasing use of real-world data with external controls for regulatory purposes now. Also involving the use, interestingly, of artificial intelligence and the development of so-called federated learning systems that leave the data within the hospital firewall, and only the algorithms that are related to the federated learning systems travel. And they are trained at the different clinical sites involved in the trial. This also has the advantage of safeguarding patient privacy. Now, to end with my talk, we clearly had a fascinating workshop on what I would say somewhat futuristic therapy that ended in a discussion on futuristic applications of algorithms for data assessment. Now, to summarize, here are my take-home messages from this meeting. It's quite clear that we are dealing with very complex technology where continuous improvements are being achieved. We are now into the fourth generation of CAR T cells. There are clearly manufacturing timeline challenges. It even requires an airport close to the vicinity of the manufacturer to get the drug or therapy to the patient as quickly as possible. Yet, it is also evident that T-cell therapies have proven activity, at least in hematological malignancies, and there are early hints of activity in solid tumors as well. Fortunately, regulatory pathways have been activated and are available now, both in the US as well as in Europe. Yet there are several aspects that really require our attention and really require further improvement. We can make use of master protocols for clinical trials, and they can be refined even further than they are now. We need to learn how we can use real-world data and the related real-world evidence in the way that we apply control arms for the single-arm studies. We can make use of artificial intelligence in the registries that are existing, and we need to make sure that we learn from the artificial intelligence algorithms on a continuous basis. And finally, and maybe most importantly, we absolutely need to have discussions on the health technology assessment difficulties to ensure ultimate and uniform patient access globally. It was my pleasure to summarize this for you. I hope you understand I'm not the real expert, but I'm happy to try to answer all questions you have. Thank you very much. Back to you, Ruth.